research with stem cells created by Soloman. For further information on Soloman, please click on the right-hand corner of the screen.
Soloman is in the process of researching lab-grown cells in hopes of finding cure for disease and illness, and perhaps lead to individualized treatments. Treatment targeted at a particular person rather than a particular disease.
So to give you a better understanding of these embryonic stem cells, they're pluripotent, meaning they can morph into any other cells in our body and repair damaged cells. Soloman describes the stem cells as our body's own repair kits, with the ability to repair cells such as liver, kidney, brain, muscle, bone cells, and much more.
The research Soloman is currently working on has hopes for learning from the stem cells why we get illness and diseases and to produce pharmaceutical drugs.
In 2005, Soloman and her team created the New York Stem Cell Foundation Laboratory to have a safe environment to do the research. Soloman realized that modern medical research in developing drugs and treatments are being dominated by large organizations. Because of this, a large gap was created between academic research and pharmaceutical companies and biotechs, those who are responsible for delivering our drugs.
To address this issue with new technologies and a new research model, she created thousands of genetically diverse stem cell lines, allowing us to understand the human genome, generating pharmaceutical drugs and treatments at a lower cost.
It was 1998 when stem cells were first discovered, and 9 years later, skin cells were reprogrammed with viruses and became induced pluripotent stem cells, or also known as iPS cells.
So these embryonic stem cells are not human, so they're terrific for use for modern disease and potentially for drug discovery. So this is a great idea to bring faith to society and making this beneficial for the people. Don't you agree, Kathy?
I certainly think so.
So in 2008, one of the Soloman scientists had built onto the research in which he took a skin biopsy, this time from people who did have diseases, a motor neuron disease in particular. Basically, what he did was took a healthy cell and turned it into a sick cell and replicated the disease over and over again. It was an extraordinary moment as it was the first time they had a model of disease from a living person in a living human cell.
At this point, Susan's team can now understand what was causing the disease.
This is really extraordinary because they have the ability to replicate many diseases in a dish and see what goes wrong with the cell before a patient could even have symptoms.
What Soloman hopes for is the ability to use human cells to test for drugs.
The modern system used for testing drugs is problematic, taking on an average 13 years for just one drug to become successful. However, the use of human stem cells has the ability to create the cells such as live. Takes an average 13 years for just one drug to become successful. However, the use of human stem cells has the ability to create a cell such as live motor neurons to test for drugs.
To do this, we wouldn't have to wait 13 years for a drug to come into the market and realize it might be unsuccessful or might even harm people.
The promise of stem cell research is relevant to society because, as Soloman says herself, at some point in our lives, all of us or people who we care about become patients.
So that's it for today's episode. If you would like to write an opinion on what you thought of today's discussion, please tweet us at Teddy Talks. Thank you for tuning in. Catch you next week when we'll be talking about glow-in-the-dark bunnies.